Good morning, everyone, and thank you for joining me today. I'm excited to present to you on the topic CMA, CPP, CQA for sterile dosage forms. My name is Dr. S.K. Singh, and I hold a PhD in this field. In this session, we'll explore the key concepts and standards related to sterile dosage forms, focusing on the critical roles of CMA, CPP, and CQA. We'll cover their definitions, importance, and how they contribute to ensuring the quality and safety of sterile medicines. Let's get started and delve into this important subject. Let's start by exploring some key concepts that are fundamental to sterile product manufacturing. First, we have CMA, or critical material attributes. These are the key properties of raw materials like the active pharmaceutical ingredient, excipients, water for injection, and packaging components. These attributes can significantly impact the quality of the final product. For example, in sterile products, characteristics such as purity, bioburden, endotoxin levels, and particle size are especially critical. Ensuring these are within specified limits helps maintain product safety and efficacy. Next, we have CPP, or critical process parameters. These are the process variables that we must control carefully throughout manufacturing to ensure consistent quality. In sterile production, this includes parameters like sterilization temperature and time, filtration integrity, filling speed, and environmental conditions such as cleanroom classification and airflow. Proper control of these parameters is essential to prevent contamination and guarantee product sterility. Then there's CQA, or critical quality attributes. These are the essential qualities of the final product that directly relate to its safety and effectiveness. For sterile dosage forms, CQAs include sterility, endotoxin levels, particulate matter, clarity, pH, and the integrity of the container closure system. Monitoring and controlling these attributes ensure the product meets regulatory standards and protects patient health. Finally, it's important to understand how these elements, CMA, CPP, and CQA, are interconnected. They work together to ensure that sterile products are consistently safe, effective, and compliant with regulatory requirements. By managing these attributes and parameters diligently, we uphold the integrity and quality of the final product throughout the manufacturing process. Now, let's look at an example involving a CMA, or cell-mediated assay, in the context of injectable solutions. When we talk about CMA, the key aspects to consider are the pH and buffer capacity of the formulation. These factors are crucial because they directly impact the stability of the active pharmaceutical ingredient, or API, and how well the drug interacts with patients. If the pH isn't properly balanced, it can lead to API degradation or cause irritation when administered. So, maintaining the right pH and buffer capacity isn't just a technical detail. It's essential for ensuring safety and efficacy. In sterile dosage forms, several other factors come into play. For example, the particle size of the API, especially in suspensions, influences how the drug dissolves and how consistently it can be administered. Solubility is another critical factor. If the API isn't soluble enough, it can affect bioavailability and stability. The pH of the solution again plays a role here, as it affects solubility and stability. Buffer capacity, which we mentioned earlier, helps maintain the pH during storage and use, preventing unwanted changes. We also need to consider excipient purity, including endotoxin levels, because impurities can cause adverse reactions or compromise the product's safety. The quality of water for injection, or WFI, is fundamental. Any contamination here can threaten the sterility and stability of the entire product. Finally, container closure integrity is vital to protect the product from external contamination and maintain its sterility from manufacturing through administration. All these factors are interconnected, and understanding them helps us ensure that injectable solutions are safe, effective, and compatible with patients. Let's take a closer look at this example involving CPP, or critical process parameters, in sterile filtration. When we talk about the case involving a filter with a pore size of 0.22 micrometers, The primary goal here is to ensure microbial removal. This pore size is standard because it effectively traps bacteria and other microbes, helping us maintain sterility. However, it's important to remember that applying excess pressure during filtration can pose risks. Too much pressure can damage the filter, compromising its integrity and potentially leading to contamination. So, managing the pressure carefully is crucial. Now, in sterile manufacturing, several other CPPs come into play. For instance, sterilization temperature and time, like autoclaving, are critical for ensuring complete sterilization without damaging the product. Filtration pressure and flow rate need to be optimized to balance efficiency and prevent filter damage. Aseptic filling speed is another key factor. Faster filling can increase productivity, but must be controlled to maintain aseptic conditions. Mixing time is also important to ensure uniformity without introducing contamination. Environmental conditions, such as operating in grade A or B cleanrooms, are essential to keep the process sterile. And when it comes to lyophilization, parameters like freezing rate, primary drying temperature, and secondary drying time are all tightly controlled to ensure product stability and quality. In essence, each of these CPPs plays a vital role in safeguarding product sterility and quality throughout the manufacturing process, connecting to our overall goal of consistent, high-quality biopharmaceutical production. Let's take a closer look at this example of a critical quality attribute, or CQA, specifically for an injectable product. In this case, the key CQA we're focusing on is particulate matter. Now, particulate matter can be a serious concern because it can cause embolism, which is a blockage in blood vessels, so it's critical to keep these levels very low. That's why strict pharmacopoeial limits are in place to ensure safety and efficacy. Moving on to sterile dosage forms, there are several other important CQAs to consider. Sterility, for example, means that there's no microbial growth in the product, which is essential for patient safety. Endotoxin levels also matter. These need to be pyrogen-free because endotoxins can cause fever and other adverse reactions. Other CQAs include the presence of particulate matter again, the pH of the solution, which affects stability and compatibility, and the assay, which measures the drug content to confirm proper dosage. Clarity of the solution is important for visual inspection and patient confidence. And finally, container closure integrity ensures that the product remains sterile and protected until use. All these attributes are interconnected and vital for maintaining the overall quality, safety, and effectiveness of sterile injectable products. They highlight just how comprehensive and rigorous our quality control must be to safeguard patient health. Let's take a closer look at the relationship between CMA, CPP, and CQA. So, starting with CMA, or critical material attributes, these are the specific properties of raw materials that can impact the final product. For example, API solubility is a key CMA because it influences how well the active pharmaceutical ingredient dissolves. Next, we have CPPs, or critical process parameters. These are the variables during manufacturing that can affect those critical material attributes. For instance, mixing time and temperature are CPPs that can influence the solubility or other properties of the API. And finally, CQA, or critical quality attributes, are the final product characteristics we want to ensure are within specifications. In this case, the clarity of the solution is a CQA that depends on how well we control the CMA through the CPPs. To illustrate further, consider WFI quality as a CMA. The sterilization cycle then acts as a CPP that impacts the sterility assurance, which is our CQA. The key takeaway here is how these three elements are interconnected. Controlling CPPs helps us manage CQAs by influencing the critical material attributes. This relationship is fundamental to ensuring quality and consistency throughout the manufacturing process. Now, let's take a look at some of the essential tools we use in a risk-based approach. One of the key methods is FMEA, or Failure Mode Effects Analysis. This is a systematic way to evaluate potential failures in our processes and understand their impact, so we can prioritize our efforts effectively. Another important tool is HACCP, which stands for Hazard Analysis and Critical Control Points. This approach helps us identify critical points in our processes where hazards could occur and implement controls to prevent them. A crucial step in this approach is to identify high-risk parameters, those factors that, if left unmonitored or uncontrolled, could lead to significant issues. For example, one high-risk area we often focus on is aseptic filling contamination. This is critical because contamination during this step can compromise the entire product. By adopting a risk-based approach, we don't just react to problems, we proactively identify and mitigate risks before they become bigger issues. This strategy helps us enhance product safety, ensure quality, and maintain regulatory compliance across our operations. Let's wrap up with the key takeaways. CMA, CPP, and CQA are absolutely critical when it comes to sterile products. These qualities directly impact patient safety, which is always our top priority. Achieving and maintaining these standards requires strict control over the environment and processes to prevent contamination and ensure product integrity. Importantly, these measures are not just good practice. They are essential for regulatory compliance with agencies like the FDA, EMA, and ICH. By adhering to these standards, we not only protect patients, but also uphold the quality and reliability of our sterile products across the board. For further support, please don't hesitate to reach out. We're here to help you with any questions or additional information you might need. Just contact us through the details provided, and we'll be happy to assist you. Thank you very much for your attention and engagement throughout this presentation. We truly appreciate your time and interest.